Hello guys, in this lecture we're going to be talking about the cell structure known as the cell membrane. The cell membrane isn't technically an organelle because organelles are typically considered the internal membranes, and the cell membrane, or the what we can call the plasma membrane, obviously that is the external membrane of the cell. That's the membrane of the cell that distinguishes the edge of a cell, or it distinguishes the inside of a cell from the outside of a cell. This lecture we're going to be talking about the structure of the cell membrane first, how the cell membrane is put together, and then we'll be talking about a specific function of the cell membrane. Now, the cell membrane has many different functions. In fact, in our cell communication unit, we'll be focusing on communication as a function. But in this discussion, we want to focus on the function of transport. How cells are able to transport materials to and from each other, or from the inside of a cell to the outside of a cell, or vice versa. So we're looking at transport and transport of materials as the function, which is what we're going to be talking about today. This is a diagram of a cell membrane. I want you to pause the video and be able to identify, come up with a list of all of the things you see here, and put those things into categories if you can. So pause this and take a look. Throughout this lecture, and hopefully throughout most of our lectures, what I'm going to do is I'm going to ask you to reflect on what we've just seen. So for example, I asked you just to pause and come up with a list of things that we see in that image. So my first question to you, my first reflection question to you, to verify that we're following this lecture and making sure that we understand, is what are the parts of the cell membrane? What parts did you see? Now challenge yourself, not only, of course you've just listed them, but challenge yourself not to even look at that list. Challenge yourself just to recall immediately. What kinds of things did you see? Hopefully you've given it some thought, and we can identify a few things. First of all, we saw lots of proteins. Proteins are an important part of cell membranes, and those were the big purple things that we saw. There are different kinds of proteins that we'll distinguish in a bit, but proteins are what we saw. You may have also noted that we saw some cholesterol. Cholesterol sounds like a bad thing. We always hear it on the news, or we read articles about cholesterol. We hear it in commercials that we need to lower our cholesterol, and that may be true, but cholesterol is a necessary thing for cells to function properly. So in moderation, of course, cholesterol is a good thing, and it is absolutely vital to cell membrane function. So we see cholesterol. We also see these things called glycoproteins. Now we see glyco and we see protein, and we should recognize the parts of this word. Glyco meaning sugar, or glyco meaning glucose, and obviously protein meaning protein. A glycoprotein is a modified protein that has some carbohydrates attached to it. And these have specific functions with regards to cell communication. We're not going to talk a lot about these yet. We see some cytoskeleton associated. Again, we talked a lot about cytoskeleton the other day in class. We should know what the function of cytoskeleton is. And we also see some extracellular structures as well that we're going to talk about in a little bit. So here we go. We're returning back to our diagram. So hopefully we've recalled and we can identify then some of these parts. Again, we see some peripheral proteins and integral proteins. Keywords here are proteins. We'll talk about what peripheral and integral mean in a second. We see some carbohydrates, all right, strictly carbohydrates. Sometimes we see glycoproteins, which are carbohydrates attached to proteins. Notice that the green polymers here are those carbohydrates, and when the green polymers are attached to these purple globular structures, those purple globular structures are proteins. Okay, we see some other extra fibers of the extracellular matrix, which would be made of cytoskeletal materials. We also see cytoskeletal materials on the inside of a cell, as well as cholesterol. So take a good look at the structure of a cell membrane. We're going to break it into parts in a second. But this is, when we look at the cell membrane is a really complex structure. And in fact, it doesn't do a cell membrane very good service if we just look at a still picture. The reason for that is this cell membrane is in many ways fluid. What I mean by fluid is that those proteins and those glycoproteins and the carbohydrates, they're floating. They're floating throughout this this sort of semi-fluid material. What we have to know is we haven't really talked about these gray molecules yet. We see a bunch of these little circles or these little dots. And those dots are having a very, very important function. We've already talked about them in this class already, whether you know it or not. But these little molecules are able to be squeezed through, just like you'd be able to push your hand through, maybe, for example, a glass of sand. You can push your glass in between the sand particles or the sand crystals. And you can move your hand in sort of a fluid motion. The same thing is going on here with these proteins and other molecules that are suspended in this membrane. So those gray molecules that we saw in the previous image, those are phospholipids, and this phospholipid should be familiar to you now. Remember, phospholipids are a form of lipid, and just like any lipid, it has a structure like a lipid. We see a glycerol backbone, we see fatty acids that are made of hydrocarbons, and those hydrocarbons, of course, are nonpolar and therefore hydrophobic. So again, we see nonpolar and therefore hydrophobic tails. But this is a phospholipid, and the first root that we see here, phospho, refers to this functional group, the phosphate group. Okay, we know that a phosphate group is PO4 with a -3 charge. So it's highly negative, it's highly charged, and because it's charged, that phosphate group makes that part of the molecule very polar. And since it's very polar, it is also hydrophilic. So we have a polar hydrophilic head, nonpolar hydrophobic tails. We have a molecule now that is, oh, this word comes up again, amphipathic. Amphipathic. It has a hydrophilic part, hydrophobic part. And like most amphipathic molecules, just like detergent or soap molecules, the hydrophobic tails, when suspended in aqueous solution, when suspended in a water-based solution, the hydrophobic tails like to hide from the water. They're afraid of the water. They will hide from the water and point in towards each other. And so what happens in our cell membrane is we get a bunch of these phospholipids together, and the phospholipids arrange themselves or configure themselves in a way that we create two layers of phospholipids. In fact, we create what we call a phospholipid bilayer. Of course, the root bi means two. It's a double layer of phospholipids. And if we can look at this double layer of phospholipids here, we see that the nonpolar hydrophobic tails are pointed in towards the center, and the polar heads, the hydrophilic heads, are pointed away towards aqueous solution, towards the water. They're attracted to the water. And so we get this really neat configuration of phospholipids that completely surrounds a cell and creates this barrier, a barrier between the two aqueous solutions. It makes a separation between the inside of the cell and the outside of the cell. The separation exists because of the hydrophobic region of our phospholipids. Because it's hydrophobic, water and the hydrophobic molecules, or the hydrophobic parts of the molecules, don't want to mix, and a barrier is created. This is going to be very important for a cell membrane's ability to regulate what goes in and what goes out of a cell. So quickly, here's another image of a phospholipid bilayer, where we see our phospholipids we see many phospholipids in configuration where our hydrophobic tails are pointed towards the center and our hydrophilic heads are pointed away from the center towards aqueous solution, towards the extracellular watery environment. Key is watery, the key is aqueous, and our cytoplasm, which is also a watery environment. And so we see our phospholipid bilayer here. Keep in mind, like I said, this phospholipid bilayer is not rigid. It's not like a wall that you can't pass through. And in fact, things do pass through it, and furthermore, the phospholipids are in some ways fluid. They move around relative to one another. They're always moving, just like all molecules are always moving around, bumping into one another, and so forth. So to quickly reflect again on what we've just talked about, I ask that you draw a phospholipid bilayer. In this class in particular, when we talk about mechanisms, when we talk about structures that we may or may not be able to physically see, it's important for us to be able to draw these structures. Because if we can draw and then, of course, label these structures, then that tells us that we have some kind of image in our head that we can apply, that we can work with. Too often I find that students who don't do very well in the class don't have a very good ability to visualize and to draw. So if we're able to draw, that allows us to start to apply some of these concepts. So what I'd like you to do is to draw a phospholipid bilayer, and I'd like you to label the hydrophilic area and the hydrophobic area. Go ahead. You can pause the video. So we've talked about this phospholipid bilayer. Again, that phospholipid bilayer has hydrophilic heads on either end and hydrophobic tails pointed towards the middle. Again, that that phospholipid bilayer creates some kind of barrier that prevents things from, or that regulates substances from moving into and out of a cell. But importantly, the cell membrane is not just made of phospholipids. What we have to recognize is that there are so many proteins involved in the cell membrane. We call these membrane proteins, understandably. And we have two different kinds of membrane proteins. We see these integral proteins and these peripheral proteins. The integral protein is this protein right here in particular, and the peripheral protein is the protein that is right here in particular. So you can see there's a difference. Try to identify the difference that we see in these proteins. If you've observed carefully, you can see that the integral protein is a protein that passes through the entire phospholipid bilayer. You're going to see an integral protein on the outside of a cell membrane as well as on the inside of a cell membrane. On the other hand, an integral protein is usually only seen on one end or one side of the cell membrane. So for example, this peripheral protein is only seen on the intracellular side of the cell membrane, on the inside of the cell. These peripheral proteins will have very important functions, as well as the integral proteins, with regards to cell communication, as well as transport as well. So again, here we are asking you to reflect on what we just said and what we just saw. Ask yourself and try to answer the question in your own words, in your notes. What is the difference between integral proteins and peripheral proteins? Specifically, structurally, what is the structural difference? Pause the video and answer the question. After you've answered that question, I'd like you to again be able to reflect and try to visualize what we're talking about. So, after you've described the difference between integral and peripheral proteins, I'd like you to add to your phospholipid bilayer one integral protein and two peripheral proteins. Keep in mind that peripheral proteins do not have to be associated necessarily with integral proteins. They don't have to be touching if there's not a possibility. All right, so by now we should have a pretty decent understanding of the structure of the cell membrane. We know that the cell membrane is made up of phospholipids, and specifically a phospholipid bilayer. And inserted into that phospholipid bilayer are different kinds of proteins, both peripheral proteins and integral proteins. Now, of course, we've only talked really about the proteins and the phospholipids, but we also know that there are glycoproteins associated, as well as carbohydrates, cholesterol, cytoskeleton. Those latter structures that we've just mentioned have less to do with cell transport than the proteins and the phospholipids. So at this point in time, we're sort of going to ignore them. We're not going to pretend they don't exist, but we're going to let them be for the moment. At this time, we want to talk about two methods of cell transport. We'll call them diffusion and osmosis. And when we talk about diffusion and osmosis, we're talking about transport, of course. These are specific kinds of transport. That's an S, of course. Specific strategies of transport. Again, when we're talking about transport, we're talking about how cells regulate what goes in and what goes out of them. So we're talking really about how molecules move into and around cells. From our inquiry activity in class, we should have been able to deduce what diffusion means and even what osmosis is and means. But this is a quick diagram of diffusion. What we see here is that there is a membrane, a lipid bilayer or a phospholipid bilayer that's right here that regulates what goes across it. And so we see these hexagonal molecules, and these hexagonal molecules start in the extracellular space. In fact, they are entirely in that extracellular space, and intracellularly, inside the cell, there are none of these molecules. So we would be able to say that there's a high concentration of these molecules outside, and a low concentration or a low crowdedness of these molecules inside the cell. And by diffusion, these molecules like to spread out. They like to move from an area of high concentration to an area of low concentration. And as you can see, they can fit through, or they can pass through the cell membrane. They can pass through the lipid bilayer. Only certain molecules can pass through the lipid bilayer, and we'll talk about that in a second. Eventually, they're going to continue to pass and move towards the inside of the cell, until we have an even concentration of these molecules on each side. What we've just experienced is diffusion. The diffusion, or the spreading out of molecules from a high concentration to a low concentration, until an equilibrium is achieved. Now, diffusion doesn't have to take place across a membrane. Diffusion can take place through any molecule that's spreading out from a high concentration to a low concentration. Just like we saw in the food coloring. When you drop a drop of food coloring, we have very highly concentrated molecules. Let me write it in black, for example. Highly concentrated molecules in a certain space, and outside of that space, we see very low concentration of those red food coloring molecules. Eventually, they'll move from high concentration to low concentration, and they'll spread out around the water so that eventually an equilibrium is achieved where there is no high concentration or low concentration. In fact, it's an evenly distributed concentration. It's important for us to be able to connect these concepts to things that we've seen before. And I know that we've seen plenty of examples of diffusion in the past, whether they are actual examples or even figurative examples where something spreads out in a sort of a symbolic or figurative way. So what I'd like you to do is, in your notes, to be able to describe another time when something spread out like the food coloring did, or spread out like those molecules that we saw in our inquiry experiment. So pause this and take a second to write those down in your notes. Here we have a definition of diffusion that you can access at any time. Diffusion is the movement of molecules from an area of high concentration to an area of low concentration. Very simple. But if we continue to remember this, and I know we've talked about this during sophomore year, but again, diffusion, let's imagine a hill with a boulder on top of it. That boulder wants to move from a higher area, we call that high concentration, for example, to an area of low concentration. And it does it with an absence of energy input. In fact, if this boulder were just simply to be let go, that it will still move from high concentration to low concentration. You don't have to input energy to move that boulder because metaphorically, gravity will do the work. As a result, we can say that diffusion is a passive process. And when I say passive, I mean that there is no energy required. Okay, no energy in the form of ATP. We'll be talking about ATP in a bit. Diffusion will occur, or diffusion will take place until a dynamic equilibrium is reached. Now, keep in mind, equilibrium sounds like equal, and that's true. But I've also included the word dynamic. So for something to be dynamic, that means there's still movement. Just because molecules have reached an equal concentration across a membrane, for example. So let's say we've got a membrane and we've got some molecules on the outside of the membrane, and they reach an equilibrium on the inside of that membrane. Well, that's about equal in terms of concentration. What a dynamic equilibrium means is, what it doesn't mean, first of all, is that these molecules stop moving, that these molecules stop crossing the cell membrane. What really happens is that a dynamic equilibrium is the definition of it is the rate of molecules moving into the cell is equal to the rate of molecules moving out of the cell. So that is to say, the rate of molecules moving into the cell. Molecules are moving in, but just as many molecules move in as move out. So for example, if this molecule here were to move in, then if it was in equilibrium, as it moves in, there's probably another molecule moving out. And if there's another molecule moving in, we have a second molecule moving out. So for every one molecule moving in, we have another molecule moving out, so that we have no net change. Delta is change, no net change in concentrations. Okay, so a dynamic equilibrium means that there's no net change in concentrations of solute or of molecules. And so the membrane that we saw in the previous couple diagrams, as well as the filter or the screen that we saw in class earlier, we can describe those things as being semi-permeable. Semi meaning partially, permeable meaning able to pass through. So it's molecules or membranes are semi-permeable when only certain molecules or certain substances are able to pass through, but others are not able to pass through. Usually molecules are permeable because of their size. That is to say, some molecules are small enough to fit through the pores or the little holes of our cell membrane. Again, remember that the phospholipids in a cell membrane are fluid, and because they are fluid, they're sort of moving all around and they can fit things through them. Okay, so size is a big condition for permeability or impermeability. Okay, but not only does size help distinguish which molecules can pass through or not, a lot of times polarity can decide. If a molecule is polar, it usually will not be able to. It is usually not permeable to a cell membrane. Charge as well. Okay, so polarity, charge, size. These are things that make molecules either polar, or make them either permeable or not impermeable. For a cell membrane, usually small non-polar molecules are permeable to cell membranes. So molecules, for example, like carbon dioxide or oxygen, obviously water, though that water is polar technically, but these are small non-polar molecules that are easily able to fit through the cell membrane. If we take a look at this diagram, I asked the question, this membrane is permeable to which color molecules? Well, we have to simply understand the term semi-permeable or permeable, and we have to ask ourselves which molecules can pass through. That is to say, which molecules can pass through the pores of this membrane. Well, certainly the criteria here is size, of course, and so we can imagine that the molecules that are small enough will be able to pass through. So I imagine, for example, this green molecule will be able to pass through, this purple molecule, this yellow molecule will be able to pass through. Certainly this red molecule will not be able to pass through, and maybe even not this bluish molecule. Certainly, again, based on the simple